Okay, so we're going to get back to our program with another great development for the Philly life sciences market this year, one of the signature accomplishments, and that is talking to 2 of the 6 or 7 companies that have raised in excess of $35 million, which in this investment climate, as you all know, is no easy feat. So I'm excited to hear their story and their journey of how they went about being successful here. So we have John Pennant, the partner in charge of the National Technology and Life Sciences Group for EisnerAmper, moderating the discussion. Zheng-Ho Chao, Chief Scientific and Medical Officer of Lattice Therapeutics, and Josh Muntner, Chief Financial Officer of IMVAC. So let's welcome them.
Good morning, everyone. Boy, those lights are bright, huh? Right in our eyes. So welcome. Again, my name is John Pennant. I lead the Technology and Life Sciences Industry Practice at EisnerAmper. I'm also a member of the Mid-Atlantic BioAngels. It's my pleasure to be moderating our discussion here today. We're actually going to start off a little different than some moderated conversations. We're going to have each of the 2 companies walk through a brief slide deck to tell a little bit of the story about the company, where they've come from, what they're doing, some of the technology that they've put in place here, and then we'll follow up with some questions on the back end there. So I don't know, do we have an order which one goes first, or?
I'm going first.
You're going to go first?
Yeah.
Do you have the clicker? Do we need to get the clicker?
Yeah, I got it.
Okay. We'll see which presentation comes up first.
Perfect. Yeah, so nice to be with you all today. Thank you. My name is Zheng-Ho Chao. I'm the CSO, CMO of Lattice Bio. And just to give you a little bit of background on Lattice and where we came from and where we hope to go. So this is a Philly-based story. This is a company that's founded on the science that comes out of the laboratory of Dr. Beverly Davidson at CHOP. As many of you know, Bev was the co-founder of Spark and has basically been on the scientific advisory board of every gene therapy company that has been in, has learned a lot of things to do and what not to do. But in a nutshell, developed some novel technologies which generated some capsids, which are really game-changing for gene therapy. Our leadership includes Peter Goroshin, who is a serial CEO, entrepreneur, biotech entrepreneur based in Philly. Myself, I am based in Boston, but spent a number of years working at Merck in North Wales, living in the Philadelphia area, and then most recently in Novartis. And then John Connolly, who comes with a background in a number of different biotechs. And our funding, we have 2 lead funders, which are 8VC and DCVC. And pertaining to the discussion we were having this morning, these are our tech bio and biotech investors. So they're our lead funders. We also have an investment from Samsung and some other funders, including Ben Franklin Technology Partners, again, emphasizing our ties to Pennsylvania and the Philadelphia area. So a little bit of our footprint in Philadelphia, and I should just say that our R&D is really based in Philly. Again, the foundational science was at CHOP. 3 of our Lattice staff, our scientists, are actually alumni from the Davidson lab. So we have an ongoing, very productive relationship with the Davidson lab in that they're doing a lot of work on behalf of Lattice under the auspices of a sponsored research agreement. We ourselves have our wet lab based in the CIC building, and thanks to the CIC staff, because I got a nice ice scraper from them today. And so that is the heart of our R&D operations. We do have another building another office that we've opened up in Boston, where much of the management team and our clinical development teams are sitting. But we are our lab efforts, our wet lab, are certainly based in Philadelphia. And we think that this kind of dual location serves us really well for right now. Just a brief summary on our technology. AAV is adeno-associated virus, and it's the predominant way to deliver gene therapy. Gene therapy conceptually is pretty straightforward. For those diseases where you understand the genetic basis, if there's a missing gene, you can replace it. Or if there's an overactive gene, you can kind of decrease the activity of that gene. The issue, especially for brain diseases, central nervous system diseases, is that these AAVs basically don't get into brain cells. And if you can't deliver it, it's not going to work. And so the innovation here is capsids that get all over the brain, and this is really unprecedented. What this means is that with our engineered capsids, we are able to get into the brain cells, which you'll need to do to fix neurologic diseases, but at very low doses. And the doses have everything to do with the amount of manufacturing and also the adverse events that a patient might experience. This is a schematic of how this is all done, but basically we start with the CNS disease in mind, and the 3 rules of neurology are location, location, location. So you have to really understand which parts of the brain you need to deliver that gene therapy to. We start with those anatomical locations in mind, and then engineer naturally occurring AAV capsids and create a huge amount of diversity at the outset. And then from that point on, after we created tens of millions of different flavors of AAVs, we then do a massively parallel, unbiased screening of which ones get to those areas that we've decided are really important for the diseases. Built into this whole process is the manufacturability, which is a huge issue for AAVs. So simply, if these capsids don't get manufactured, they don't make it to the next round. And we do this using the model that's closest to the humans, which are non-human primates. Again, this is random peptide insertions. This is where the huge diversity is created, but then we solve it in biology, in the brains of these animals, which is the closest we're going to get to humans. And then the field has moved away from trying to do this capsid evolution in rodents and then hoping that they work in non-human primates and then to humans. We're starting as close as we can to the human. So we have a couple of lead programs just to show you how far we've come. One is named for the capsid, which is called DB3. It's called Deep Brain 3. And not surprisingly, when you administer this attacks the deep structures of the brain. And that's important for certain diseases, especially Huntington's disease. And so one of our lead programs is a Huntington's disease program using this deep brain capsid. We have another program which it transduces the ependymal cells of the brain. I'm sure you all remember from your neuroanatomy that the ependymal cells form the lining of the ventricles, but these cells produce CSF, the cerebrospinal fluid, and secrete proteins into the CSF. So by transducing these cells, we can convert these cells into protein synthesis factories. And we're using this capsid to take into a pediatric neurodevelopmental disease called CLN2 Batten's disease. Just one example here of our DB3. On the right in the green is a conventional capsid that has been used, which is AAV5. And here it's encoding a fluorophore. This is an NHP brain. And what you see is you just don't have very many cells labeled. Whereas in the teal, which is the DB3, we're getting tremendous coverage of the cells. So this is a huge, difference in the number of cells transduced. Again, if you don't get it into the brain cells, you're not going to fix the brain cells. We also have, this is kind of remarkable the first time I saw it. You can inject these viruses in one part of the brain and they swim to other parts of the brain. So all these different locations are numbered. They're all from a single injection, and Neuroanatomically, these are locations that are very far apart. So because of the inherent natures of these capsids that we've selected for, they will distribute throughout the brain, including, by the way, all the brain areas that are really important for Huntington's disease. So this is just another example of the EP+. And remember, the ependymal cells are lining the lateral ventricles, and you can see on the left that we basically can transduce all of the ependymal cells. It's EP+ because it does not only transduce the ependymal cells, but also some neurons, so EP+. And so we think that this is a way to get very high levels of your therapeutic protein. We've seen this for our lead compound, LTS-101, on the left, and we can measure the amount of the missing enzyme, and there are very high levels, up to 100 nanograms per mil, whereas previous efforts using other AAVs are only getting about 8 to 10 nanograms per mil. So by using these cells, transducing these cells, we're able to get high therapeutic levels. And so we are headed towards the clinic. We have later this month a pre-IND meeting with the FDA. And just, this company is just 2 years old. We are farthest along with CLN2, but we're also advancing our Huntington's program, heading into the clinic with both of these. It's kind of an interesting challenge for a startup to have clinical programs, because then you're bringing on staff that are not just in the lab, but regulatory, clinical, statistics, et cetera. So our staffing is very spare, I would say, which is a design principle. And to make that happen, we've raised 54 to this point and hoping to get additional funds. And I think that's it. So let me pause there and.
Yeah, great. So just a quick question before we turn it over to Josh there. So how do you get the viruses to get to the selective cells you need them to get to? So you're explaining how they get all over the place. How do they land where they're supposed to land?
Yeah. So the question is, how do you get the viruses to get into specific cells? And there are companies that are trying to engineer this by understanding the chemical nature of each cell, and then design viruses that will go specifically there. And there's been some encouraging results in that. We haven't done that. We've created a lot of diversity right at the outset, and just see what the winners are. So why they're going or swimming to different locations, we don't actually know. Okay. But I kind of liken this to the early days of web search, where there were, Lycos and Alta Vista were like looking at different websites and rating their quality, and then Google comes in and says, look, we don't care. We're just going to see which the winners are by numbers. Yep. So I think the key is that we have massive diversity up front. Everything's barcoded. It's a very heavily bioinformatically driven system. So we can process in parallel for millions and millions of capsids to see what the winners are.
Great, terrific. Thank you very much. Josh, you want to introduce your company a little bit for us?
Sure, thank you. My name is Josh Muttner. I'm Chief Financial Officer of Imvax. We're a private company based here in Philadelphia. I had had a career in investment banking, working with biotechnology companies, many of whom were Philadelphia or in the Mid-Atlantic region, helping them raise capital, do strategic activity. Worked for some time even at a Philly bank, Janney Montgomery Scott, and went over to industry, and this is now my third role. Been at 2 public companies, now at Imvax as a private company, and very excited with what we're doing. Since I joined about 3 years ago, we have moved our program forward quite dramatically. And I think I have some slides that may pop up. There we go. Very nice. We have a tremendous technology platform that we've in-licensed along with our co-founder, who is on faculty at Thomas Jefferson University. In-licensed the technology from TJU. We share IP with TJU. And we've moved this platform technology forward to treat glioblastoma primarily, as well as apply the technology to other tumor types. We were founded by a neurosurgeon, so GBM was his main focus, and that became the focus of the company. We recently fully enrolled a phase 2b study treating GBM patients, nearly 100 patients in the trial. It's a randomized controlled study. We don't know where it's, what the results will be. We'll read the data out in the first half next year, but very, very exciting time for the company. We did this because of the earlier data that had been generated in an IST at TJU, which showed very, very compelling phase 1 data, more than doubling progression-free survival and overall survival in the patients studied there. The intervention that Dr. Andrews came up with at TJU was taking what's done in standard of care glioblastoma treatment, which is craniotomy to remove the tumor. The patients recover and go on to standard of care therapy, which is chemotherapy and radiation therapy. We intervene taking some of the tumor tissue, modifying it, packing it in a chamber, and then implanting the chambers back in the patient for a period of 2 days, and turning their own tumor cell into a vaccine-like substance against their cancer. We got tremendous results in our earlier studies, and I think I share that. Oh, the colors are different than they normally appear for us, but I think they're Halloween themed here. Typically, GBM patients, unfortunately, the results are very dismal, with patients progressing in about 6.5 months, cancer growing back, and then median survival about 16 months, and that's shown on the far right side of the slide. In our study, as shown in the overall population, there was some effect, especially with respect to having the cancer not grow back as quickly. In the highest dose cohort, and then the sort of apples-to-apples comparison with that standard of care, we more than doubled both progression-free survival as well as overall survival. That led us to push into this phase 2b study, the nearly 100-patient study that I mentioned. That was enrolled at about 19 sites up and down the East Coast and as far west as sort of Wisconsin and down into Florida. So good diversity of sites in the trial. All of the tissue is processed at our facility in the Curtis building here in Philadelphia. Here's the timeline of the trial, starting the trial in 23, finishing enrollment this year, and then moving into data next year. We think the trial could serve for an accelerated approval filing in the near future. We've raised over $200 million to date, including $57 million over the last 2 years in support of the phase 2 study. And that was really just as we were enrolling patients, needed to keep the funding in the company. And we've had tremendous support by a number of investors shown on the bottom here, many of whom are actually large family offices, not necessarily the typical venture capital funds, but they've been tremendous in their support year after year with us. Here's a slide about our in-house manufacturing facility. That we built this out in the Curtis Center in Philadelphia, about 40,000 square feet. Cost was about $15 million. We did get support from the state of Pennsylvania through the RACP grant funding mechanism. Took a while to get the money. It did not fund all 15 million of it, funded 2 million, but it was good to get their support in the build out. And then this facility is GMP certified and was used for the conduct of our study. As I mentioned, we can apply the platform technology to other tumor types. So we have some ideas of where to go, some concepts in one word, but glioblastoma is where we're spending most of our time and money right now. So accomplished a lot over the last few years. I'm going to get through these slides so we can get to the Q&A.
Terrific. Thank you very much for that presentation. Let's just start off with a couple questions there. So you mentioned in your presentation there, Josh, that you had gotten an incentive package from the state of Pennsylvania, one of the grants. Had that been an important part of your strategy, either getting early scientific grants like NIH or things like that, and or federal or state grants and things like that?
We had unique sort of origins with the technology coming out of TJU. and Dr. Andrews running a phase 1B study at TJU under an IST. And so that was really prior to forming the company. A lot of the work was done by him, a lot through philanthropic means that he generated prior to IMVAX.
Okay.
Once we formed the company, we pulled together a Series A. We did use the non-dilutive funding for building out the facility.
Sure.
Otherwise, my experience is sometimes NCI, DOD, the money is useful and validating, but slow to come.
Right. Yep. Django, how about yourself? Did your company take advantage of any federal or state grant programs?
Not federal or state, but we applied for a grant from the Hereditary Disease Foundation. The HDF has been a funder of Huntington's disease research for many, many years. And recently a family came to HDF that was newly aware of Huntington's within their family. And they kind of said, why aren't there any cures? And somebody explained to them the Valley of Death. So they endowed these transformational research awards, understanding that that there are lots of good ideas in the laboratory that never make it to patients. And so we applied for this grant in conjunction. So there were 3 applicants. There was Bev Davidson as the lead PI. There's our collaborator, Leslie Thompson from the University of California, Irvine, and Lattice Bio. And with the idea that we had targets in mind, we had an experimental plan, and we got the grant. And this is the largest, so it's $1 million over 2 years. It's the largest grant that the HDF, which has been one of the major funders for HE research for decades, but it's the largest single grant that they've ever given out. There were 2 grants given out a year ago. Interestingly, we're not, as Lattice, taking any funds from that grant. That work is being done in the academic labs. Okay. Meanwhile, we have worked out the license. So our end of that is getting it into the advanced animal testing. And so that is, I think, it's a nice example of it's not necessarily all about the money. Right. Money is good, but
progress, right?
The progress and also the recognition. This is a high, high prestige grant, and I think to our mind is an illustration of kind of what it takes to take academic innovation and really bring it into something that could be a drug.
So for many companies that are working on unique diseases, there are patient advocacy groups that are focused on that. Did you find them, or did they find you? I guess I know a lot of companies would like to access, but often have difficulty finding the right connection point to make that happen.
Yeah, so I'll start and then I'll toss it over to Josh. For us, and I'm a neurologist, neuroscientist, I'm a clinician, I think our North Star is not our technology. Our North Star is patients and deploying the technology in a way that can really benefit patients. And so it's built in that we're going to really start with an interaction with the patient organizations. I would say for our CLN2 efforts, we reached out to the major patient organization. We actually collaborated with them in doing a caregiver survey in terms of, is there really a need for a new therapy? What's the tolerance of the community for gene therapy? Would you do a clinical trial? What are the biggest burdens, et cetera, which we ran in conjunction, which we helped pay for, but they executed that patient survey, which was kind of eye-opening in a very good way. Just a note with these, especially with these rare disease communities, we're trying to be very careful. So if you look up Lattice Batten's disease community survey, you won't find it, and we've kept our name off of it because I think the rare disease community will get very excited that there's a biotech that's working on their disease, only to have those biotechs kind of decide for strategic reasons we're going to stop that program. We we really wanted to be very careful at raising false expectations, and yet we still wanted to engage and find out really, what we should be thinking about right from the outset in terms of what the therapy needs to deliver for the patients. Having said that, we've met with a number of different disease communities. Word has gotten out that we've got capsids that could be relevant to their disease, and it's careful. We have to be careful about going into this because we can't say, personally, I want to cure all the diseases.
Sure.
But realistically, we can't take them all on at once, and so we're trying to understand kind of what the reality is for these patients and their families. But also to manage expectations and really not raise false hopes, because I think that in the rare disease community, there's been a lot of that's gone on.
I love the way you present your North Star there as well. So,
Okay.
Yeah, very important.
Yep.
Patients, of course.
yep. Josh?
We have spoken to some of the foundations about funding IMVACs. They generally are focused on earlier stage research, and their dollars can be really leveraged there, finding new approaches. At our stage, we're clinical, and so the dollars needed are larger than the foundations can do. We do work with a lot of them and are sponsoring some of the American Brain Therapeutics sort of walk. We were in Houston last week. There's one in Jersey coming up October 12th, I think. So we try to be out there in the community.
Terrific. Since our panel is focused a little bit on the financing side of things, Josh, maybe we'll start off with you a little bit. So you've raised a lot of money. How did you do that? Was it sort of in tranches? How did you know how much to raise, when to raise, milestone based? Give us a little bit of the thought process to how you got the amount of funds that you did, which is obviously enormous and significant.
So, of course, biotech can be a game of sort of fits and starts, chutes and ladders with all sorts of hurdles popping up. We were blessed to have raised a lot of money in 2020 when the markets were very robust, especially for biotech companies. And over $112 million was raised by the company at that time. That sustained us through a very sort of dark time, 21, 22, that has really dragged on quite a bit, and some would say that we're still in that sort of dark time. Yeah. But it allowed us to do a lot of the validation work, get the manufacturing set up, and be ready to launch the trial. And then ultimately we did have to raise money very sort of project focused around running this study. We did it a lot through relationships, getting out there, staying in front of people, and my background being Wall Street, especially in the biopharma community, I think helped the company a bit.
Sure.
So.
Yep. And when you were able to raise that money, did you have a specific amount in mind that you were looking to raise, like enough to finish your preclinical, your clinical? Was it kind of targeted to a specific amount?
Investors do like to invest to a milestone, and so I think your chart showed it very well. Here's what we're going to do with this amount of money, and so you definitely do that.
Yep. Any thoughts from your perspective on those early raises? I know you're on the science side, but
I will just agree that 2023 and 2024 has been a very tough year. We've raised a good amount of cash to keep us going for a while. But we've gotten a lot more no's than we have yeses. But I would say that the raising to milestones is really absolutely important. So kind of the question of, well, what data are you going to have by when, and how influential is that going to be for the markets? And so it's forced us to really say, okay, to plan out our experiments and to see into the future in a way that that is a little bit aspirational, but realistic. The other flip side is, on the science side, the best strategy, I think, is just keep cranking out the data. And in that regard, it's interesting. We have a relatively young crew of scientists. They're very smart, but most of them haven't been in industry. But what they all know now is the value of the data that they're creating is going to tip the scale for some investors at some point. And so we've been lucky, knock on wood, that we've been continuing to generate data, which is first and foremost advancing the programs, because we have these Gantt charts and we know where we want to get to, but also that our impressing our investors that we know where our data stack up against competitors and why we're better in this regard, et cetera. So just kind of keep on swimming and generate the data is the recipe, I think.
Right. The data will set you free, right?
Yeah.
As they say. So you referenced sort of the 22, 23, rolling into 24, kind of the dark days. Both companies have been fortunate to have a lot of funding, but I'm sure you, like everyone else, have had to think about prioritization and techniques to extend your runway and to advance and get more data with not as many resources as you were hoping for. Could you talk a little bit about what you did to help extend that runway and just to keep persevering in that tough time?
Yeah, I'll start, but I'm also very interested in Josh's answer because doing those clinical trials is so resource intensive. I'm sure you had to make some tough choices, but look, the capsids, the viruses that we have could address a number of different diseases, and I want to cure all the diseases, and I know that we can't. So really, I would say prioritization and focus is. We're also very open to, we don't want to necessarily kind of keep these capsids to ourselves. We think it makes sense to extend the reach and the impact of the technology we have to partner with other groups because we can't do everything ourselves. We're a very small group. So those are ongoing discussions, but also aside from potential partnering for diseases that we're not already in, understanding that prioritization and having that discipline and not going after everything you can think of is really the way to do it. And so the faith-based argument is that we're going to demonstrate our credibility and ability to execute, and that should, eventually when the markets do come around, we think that'll all be very virtuous stuff to point to.
Terrific.
Yeah, we have, we're currently about 50 people based around the Philly office. And we've had to make some changes since I joined in 21 till today, as we shifted from a sort of figuring out how to manufacture the product and how to run the trial and doing process development work to actually executing the trial. We made a big shift in the organization and had to move resources around, and some people had to leave the company as a result. Very, very tough choices to make. The last year and a half has been around running the clinical trial, executing that. And with the conclusion of enrollment, we had to, again, make some choices around resources we'd have at the company. And these are very, very tough decisions.
Yep. I have a few more questions, but I will open it up to the audience if there are any questions that you have, if I can see anybody out there. All right, so just while we're queuing up there, Josh, maybe I'll tap into your former investment banking background there and give us your thoughts on what 24 is going to look like the end of the year here going into 25. Give us your crystal ball here in the Crystal Tea Room.
So there has been a resurgence in IPOs recently, with I think 4 of them or so going last couple of weeks.
Yep.
A couple on the docket right now.
Couple of reverse mergers close. Yep.
Yeah, the reverse mergers are a tougher way to go, but it is a source of financing for companies to get out there. But the hope springs eternal that 25 will turn a new leaf post-election, post-interest rate cuts. There's certainly been plenty of money raised in the venture capital community. There's a lot of money on the sidelines. Some mega funds with Arch Ventures or Bain Capital, as well as a lot of new funds forming.
Yep. Yeah, so I mean, we've seen companies that have, I'll say over the last year, have told us, like from sort of like the accounting and auditing side, like get ready to do the audit so when we can go IPO, we'll go really quickly, but don't actually start yet until we're like sure that the money's there and we're going to be able to get it done. Which is always great for our business because then we get that phone call like on a Tuesday morning here saying, can you have the audit for the last 2 years done by Friday? I know we talked about this 2 years ago, but that's no problems, right? So it's inevitable, but that's what happens. Yep. Any other questions there? Otherwise, I'll keep going with a couple other things.
Thank you. Just a quick question. You were talking about changes in personnel of the company as you were moving into the clinical. What are your thoughts about the profile of the company individuals and how is it communicated to the investors at different stages of the company progress? Because oftentimes people who can discover are different from people who can develop and different from people who can commercialize. And the investors look not just at the technology, but as to who is on board to actually drive things forward. So what are your thoughts in terms of optimal talent within the company towards both achieving your milestones, but also towards communicating your capabilities to the investors?
So, there's always a leadership team that's involved, and usually the team leaders, like our chief scientific officer, was not part of the team that maybe got moved out, but some of the people working with him did, as we had to refocus the organization from the research to the development side. It's important to still, be publishing, always be publishing, and so, it was a matter of sort of emphasis and sort of how much we were going to have in that side of the organization versus the development side of the organization. Does that help? Okay.
Anybody else?
I know we're getting relatively close to time here, so maybe I'll ask each of you to kind of give us a final thought on what do you wish you would have done differently or maybe earlier in your progress to continue to advance? Obviously, both companies have made tremendous progress and should be proud of your accomplishments, but what do you wish you could have done better or earlier?
And I'll start. I wish we had more money. I wish we had more money.
Shocking question.
And it's simply because I think we could have gotten more stuff done. But I'm an optimist, and I think actually, as a startup, to be very careful with our dollars and to make sure that everyone in the company understands the value of what they're putting in and why investors matter and why milestones matter has been, I think, has set us up really well. I do see a time when funds will be more available. I think that's coming. Not the least of which is, big pharma, such as my former company, Novartis. A lot of these companies have cut their pipelines, but eventually they all will need to have approvals. And I think something's got to give. And I think the cutting the pipelines is anticipatory towards kind of in-licensing more opportunities. So I think biotechs who are able to advance, again, with data, their programs should be in a much better situation, hopefully in the near future.
It's always more money, but Invax was blessed with a very large Series C round with a lot of support in 2020 that actually covered up for a lot of difficulties that the markets presented, but we also sort of created our own difficulties in manufacturing and problems we had to work our way through. An answer might be, we're very focused on glioblastoma, and maybe having a second product would be a nice idea, but we've seen in this market that a lot of the biotech companies live and die by one product. Even if they have a second one, if the first one fails, they're dead man walking. Yep. So I'm not even sure if that's the right answer.
Yeah, and we've seen some companies using maybe NIH grants or something like that to maybe advance a second program while putting all the investor resources into the lead program as a way to build a pipeline. But I think ultimately, so much of the value is ascribed to the lead program anyhow. It's hard to imagine.
Yeah.
Yep. Terrific. So I think we're just about at time. So I'd like to thank our panelists for your great insights.